Hello and welcome back to my channel Explaining Biology Clearly. Today we're going to take a look at the structure of amino acids, a little bit about some of the common parts that you find in all 20 different amino acids, and we'll mention how they join together to form polypeptides and how that polypeptide folds into a functional protein. And so on the screen here, what you'll see is in the middle there, three different representations of amino acids. So what you'll notice straight away is the elements that they contain. So amino acids contain the elements carbon, hydrogen, oxygen, nitrogen. And one that I haven't drawn here, which is in the R group. Now, R isn't an element on the periodic table, but it represents different elements. And in some R groups, you find the element sulfur as well. So that is written in brackets. Now, with those R groups, why have we got an R there? So as I said, it represents different groups because we've actually got 20 different amino acids in nature, and that each of them differs by their R group. So the rest of them is the same, but the R group is different. And some of them, as I said, contain carbon. On the bottom of the screen here, I can show that further. So the R group is in blue, and you can see that we've got an amino acid called glycine, and its R group is just a single hydrogen atom. Whereas valine has a more complex R group, as you can see represented here by the elements in blue. So each of the 20 different amino acids contains a different R group. That's how they differ. If we look at their general structure though, what we'll find apart from the R group is exactly the same. So we have got here an NH2 group, which is referred to as an amino group. So you can see that's common in both glycine and valine. You'll notice here the NH2 is drawn differently. We haven't shown the bonding between the nitrogen and the hydrogen. The nitrogen comes first, by the way, because it is what is bonded to the carbon, and then the hydrogens are bonded onto the nitrogen. Then on the opposite side of the molecule, what we've got here is a carboxylic acid group, COOH. So you can see over here how this can be represented just by COOH. Again, we're not seeing that bonding that's involved. So be prepared for seeing it written like that. So those are the key indicators that we're talking about amino acids. They then, of course, got the R group, which we've mentioned before, and that can be represented as in valine here, showing the exact identity of it, or it can simply be shown as an R. Got a central carbon and then a lone hydrogen sticking out from there. So I've actually drawn an identical molecule amino acid there, and then over here I've drawn it on the right hand side, I've drawn it how it may also be shown. So that's the general structure that we've got there. Now, amino acids, so they're monomers, they're building blocks. They're the building blocks for the monomers of proteins. So they've got to join together to form a polypeptide and therefore a protein. So the way in which they join together is like this. So we have the formation of a bond, which I'll draw in purple, and the bond occurs between the carbon in the carboxylic acid group of this amino acid to the nitrogen in the amino group of this amino acid. So that is our bond. Whenever we are asked to label a bond though, we also show the groups that are involved. So this would be our bond here, and the identity of that bond is a peptide bond that has formed. But in order for that bond to form, we need to lose water. So we lose this oxygen and hydrogen from the carboxylic acid group and we lose the hydrogen from that amino group there in the formation of that peptide bond. Now that is that loss of that OH group has formed water, and so that is a condensation reaction that has formed that peptide bond. We have formed a bond and we have lost a molecule of water. So therefore we have a condensation reaction. The opposite of that would be hydrolysis. If we were to break that bond, we would need to add back water to break a bond. Hydro means water, lysis means breaking. So the opposite of a condensation reaction is a hydrolysis reaction. Now, I mentioned that we'd talk a little bit about polypeptides. So polypeptides are where we have lots and lots of amino acids joined together. So if I represent amino acids by little beads here, imagine a beaded necklace. If we had lots of amino acids joined together by condensation reactions, we would have a polypeptide. And now, before that can form a functional protein, that polypeptide will have to fold in some way. It could fold like that into a globular shape, or it could simply just fold into an alpha helix like that, but it will need to fold. And in order for it to fold, to form the functional protein that can carry out a function, there needs to be bonds forming, and those bonds form between R groups. So bonds between R groups hold the polypeptide in maybe a secondary or a tertiary or even a quaternary shape. And that's what allows it to become a protein, because it gives it a particular shape, and then the shape gives it its function, and with a function it becomes a protein rather than just a polypeptide. So bonds that hold proteins in their shape are between R groups, and that is why it is important. The order of amino acids are important, because they determine how the protein will fold, and therefore its shape and its function. So bonds are important, therefore the order of amino acids is important. And we'll talk more about how the order of amino acids are determined in future videos. I hope that's been a useful revision of the structure of amino acids. Don't forget to check back on my channel for further videos on other post-16 topics, and please don't forget to like and subscribe. Thank you very much.